Right. So,
And here we go. We're now recording. And so thank you all for being here. Thank you, GenomeX team. And over to you, Kelly, to start us off.
thanks. Great. First, I guess I wanted to welcome you all and thank you for joining today's community of practice. I'm excited that we get to highlight the GenomeX initiative and how we're unlocking the potential of genomics through structured, interoperable data. So today's presentation, we're going to walk you through our vision, progress, and how the community, this community that we're talking to today, can help shape what's next. Basically, this is our why. Genomics is about enabling better care and research through data that's computable, shareable, and trusted. So precision medicine cannot thrive on unstructured documents. We need real-time, usable genomic insights, and we get there by working together. We have a great group working together. We've already been introduced, but there's our glamour shots for everybody today that's going to be talking. I'm excited about my team that I work with here. So on the next slide, just to give you a quick outline of the flow of our presentation today, we're going to have 4 parts. I'm going to go a quick CodeX overview, look at how CodeX bridges innovation to real-world healthcare impact. We're going to meet genomics and introduce the genomics use cases and why it matters now. We're going to talk about some specific actions and updates, what's been accomplished and where we are today. And Bob and Shrikar are going to go into that in great detail for you all. And then we'll follow up with a call to action and any questions that you guys might have. On this next slide, I know a lot, BVO, you may have covered, but we believe from the GenomeX side that CodeX isn't just another healthcare initiative, but rather a movement to break down silos and make data work for people who need it the most. So while standards are what we do, the real heart of what we do is for the real world impact. So when patients, providers, researchers in the industry are all speaking that same language through a common standard, we can unlock something powerful. We have data that moves, informs decisions, and actually improves care. So that's what CodeX does. It's not about the data for data's sake. It's more about ensuring that when the moment of care arrives, the right information is available instantly, accurately, and meaningfully. So we've seen this work in the oncology and cardiovascular health side of things. Now with genomics stepping onto the stage, we have this chance to build something transformative, something that doesn't just exist in research, but actively shapes patient care. And this, we hope, is just the beginning for everybody here. GenomeX on the next slide is different because it refuses to solve genomics in isolation. While many efforts are focused on proprietary solutions, genomics is built on the belief that genomic data must be interoperable, accessible, actionable across all of healthcare, not in those silos. So what makes it my heart and why I'm truly behind this is because I think genomics really has the power to change lives, but only if we can actually use it at scale. So too often I think groundbreaking science is left on the sidelines because it's not integrated into real world clinical workflows. GenomeX is changing that by ensuring that genomic data moves seamlessly between those labs, providers, researchers, payers, so that every patient can benefit from precision medicine, not just the lucky few. This isn't just about building another platform. It's about making genomics a true standard of care by embedding it into the very foundation of healthcare interoperability. So we feel that's how we're going to make science matter, and that's how we're really going to drive impact, real impact. Next slide. This is just real quick how CodeX is scaling interoperability across all our domains. So with cancer and cardiology paving the way, and obviously founded in MCode, we're now applying those lessons and those lessons learned to genomics. So we're validating that shared standards work across those specialty systems and stakeholders. On the next slide, what's different about GenomeX? We're expanding beyond the usual participants. This isn't really theoretical interoperability, more like thinking this is data in practice. We're not just defining those standards, we're implementing a new standard of care. And that requires a full ecosystem, including all of you on the call today. The next slide, to succeed, we think we must move beyond traditional roles and siloed efforts that we talked about. Genomics is intentionally broad. We want to invite pharmacists, AI developers, policymakers, patient voices. Outside just the lab, we need to bring everybody in so we can take those standards from theory to practice and from concept to care. And finally, we have a little exciting announcement. We're excited to say that we're launching a new model within CodeX, the domain leadership team. So for GenomeX, we're going to have a strategic group designed to provide cohesive guidance across all of the genomics use cases, making sure that we are keeping our focus on not overlapping on one another and understanding how we can pull together and understand the synergies between all of the use cases that we're working on. And Bob and Shrikar are going to go into that in just a second. So this team is created of members, and has to be a member of GenomeX, and bring together their leadership voices from each use case. That way we can make sure to align goals and priorities across a domain, streamline decision-making to avoid duplication of efforts, ensure coordinated execution and shared success metrics, and create clear pathways for the community collaboration and impact. So this is really mostly about amplifying the great work that's already happening and ensuring we operate as one aligned, efficient, and visionary team. It's not structure, so it is a structure built to empower, not to override anything that's happening in each of our use cases. So GenomeX will be the first domain in CodeX to pilot this leadership approach. We hope it will be something that begins to be adapted across oncology and cardiology and any future domains. We're proud to lead the way with a model that's intentional, inclusive, and built to scale with integrity. We're excited about what this will unlock, and we're even more excited about who'll be a part of shaping it. So with that, I'm going to pass it over to Shrikar. Thank you for being here. We really appreciate all that you do for us, and so we're excited to have you here.
Thank you, Kelly. So I think before we go into my slides, I think one thing that inspires me about GenomeX, and also in practice, a lot of organizations or standard development fail, is the complexity of GenomeX. So the complexity of genomics in general. So I think there is I think it requires a combination of, personally, I believe it requires a combination of clinical bioinformaticians, clinical informaticians, and also clinicians together working to really understand what are the end-to-end needs of the precision genomic medicine delivery in the healthcare or in research, right? And not only that, but also having industry partners, the pharma companies and also academic partners who are hospitals, who run the hospitals and everything, and also the vendors, right? The EHR vendors, and other app developers within this space. I think so far, I think it's been a few years now, GenomeX is up and running, and we are able to really bring multiple expertise together and also multiple partners, industry, academic, and also pharma partners into this into standards development here. So currently, as part of GenomeX, initially we started off with two use cases. One is data exchange use case, you see it here, and also operation use case, which is primarily led by Bob Dolan. So last year or early this year, we kind of transitioned our operations use case into molecular tumor board and pathogen genomics. So I will be, next few slides, I'll be going through data exchange use case, and later Bob will present on molecular tumor board and pathogen genomics, which just started on this. Can you go next slide, please? Again, I think as the name itself mentions, data exchange is basically it's referring to the genomic data exchange. So if you look at the diagram on the right here, healthcare organizations, they order the genetic test, and it goes through the internal laboratories or external laboratories, genomic laboratories. And once the tests are ready, the results are sent back into the healthcare organization. So as part of GenomeX, as part of data exchange use case, we are using mechanisms of the FHIR as a standard, both for both for ordering purposes and also sending the results back to the healthcare organization. So one thing to keep note, if you're really new to genomics, is it is complicated, right? You have different types of variants. If you look at, you have copy numbers, SNVs, and small indels, large indels, translocations. There are a lot of different types of variant, and also different types of omics. You have methylation, you have basically epigenetics, if you think of it, and also you have RNA transcriptomics, proteomics. So there is a lot of complexity in terms of technologies and also the chemistry that you use that you obtain from the laboratories. So right now, I think again, right now a lot still very prominent is people use PDFs, to send the data or transmit the data between institutions. And as you know, those are not really effective way to do it, right? The PDFs only capture some information, not everything. So the solution here I think we've been working on with the community. It's a community solution, not something private or something, but it is just how to standardize genomic data elements for the real-time clinical decision support, and also how can we develop this FHIR-based transport methods for seamless data exchange of FHIR files and stuff. And also, main important thing is adoption, right? So right now, again, as a part of GenomeX, we have Epic, Oracle, and all the some of the EHR members. We have part of it. So I think we're adapting how to adapt to EHRs, and also not only that, but can we go beyond EHRs into pharmacies, payers, and also direct patient access. So the impact obviously is clinicians will be able to make actionable genomic insights at the right time, and that is really, really important. Not to spend too much time into going through PDFs and all those scanned images. And also for the payer perspective, I think if you look at molecular tests are really complicated, with the payer system. A lot of them are very expensive. Oftentimes you need to get pre-approval, from the insurance companies to get it done. So I think we're hoping this also can help, with the payer approvals and reimbursement. And also this aligns interoperability with industry standard like NC PDP. Can we go next, please? So genomics exchange data use case. This is very, very successful use case. It attracted again multiple institutions and companies and vendors. So this began in around 2023. We started as a pilot phase 0, and as a part of this first phase or phase 0, we did do 2 prototypes. One is the payload prototype, and the second one is the transport prototype. Payload prototype is basically initially in the phase 0, we chose somatic reports, mostly FMI, Foundation Medicine, and Tempus, and also and then ask the question: okay, this is the different elements we have in this report. Can we convert them or represent them in the FHIR architecture? So I think we were able to do it in the STU2 at the time, using in combination with ENCODE. And again, we were successfully able to map several elements, and we were able to also test them out in connectathons and other venues. The second aspect is transport prototype. Again, this is how to identify the means for transmitting FHIR-based lab report as a link within the HL7v2 messages. Again, we tried several things there, but again, this was also tested between Tempus, Phenotypes, and Epic as a EHR vendor. So again, this is based on this success in 2023. Again, this also not only successful but also attracted new partners. Like for example, Myriad Genomics came on board, and few other institutions came on board to participate in this initiative, in part of as a phase 1. Can you go next slide, please? The phase 1 is done. It started and ended in 2024. Okay, all the scoping and everything is done in 2024. Here we expanded the somatic, reporting. Some of the elements in the phase 0, we had some we could not represent or we could not, complete them. So we thought we would push into phase 1. So we worked on those, and also we also worked on germline reports also from Myriad Genetics. So it is again. This is very much again community effort and again different. We have the code developers, we have subject matter experts, everybody come together and really we're able to deliver this on time. And not only that, again, we are able to showcase this in the connectathons, both the January connectathon and September connectathon. So it attracted large number of participants in the connectathon, the HL7 meetings. So I want to give a big shout out or thanks to James Patterson, from MIDER. He was a use case coordinator for this before Kelly stepped in this year, and I think he did a phenomenal job in bringing to the people. And honestly, a lot of time my name is there, but he's the one who drove it in terms of Connectathon, preparing for Connectathon, pulling people together and executing the different venues. So anyway, I just want to thank James Patterson on this. Can we go next, please? So again, we are working on a phase 2. Phase 0, phase 1 is done, right? 2020, 2024, and 25. We began to work on phase 2 pilot. Okay, so at this point, we are in the process of defining the scope and also timelines and who are the different contributors, to make to work on this phase 2. Some of the examples we're expanding on the genomic reports is expanding to how to represent RNA-seq, or methylation, if you think of our splicing. And there are also a lot of interest in biomarkers, both from pharmacy companies and labs and clinics and stuff. Another thing is how can we transmit VCF files, right? VCF is for those of you who do not know, that is like a raw file where you store all the genetic variants. It may contain millions of variants for whole genome sequencing. Oftentimes the ordering providers, they nowadays asking for VCF files because they want to explore in the future. And again, this is we just I think Kelly is getting used to this one and bringing people together on this. And I think this is the right timing. If any of you or your institutions or your colleagues or friends have some interest in this area, I think we definitely can educate where we are right now, what we did so far, and what are the remaining challenges we have, and how are we tackling at this moment. So I think I want to plug this opportunity to welcome any new members to participate. Can you go next slide, please? So overall, I think we've been. This is not a dry modeling exercise, right? We took a concept and tried to implement in practice, and during that process, we get to know some gaps, which we never thought, or our HL7 working group did not think about, when we were trying to model the genomic data. So we were able to really, take a concept and implement it, understand the gaps, and try to fill the gaps across various pilot, phases. So I think again, just to highlight a few things here, the demonstrated impact we have is we standardize essential genomic data elements to ensure consistent reporting across healthcare. At least we showed them in the oncology and also in the germline testing. But again, there's a lot to be done. We want to expand beyond just the genomic reports, including we want to do biomarkers and other pharmacogenomics and etc. Again, this matters quite a bit. Again, genomic data. It is sometimes it could be just a particular condition a patient may have, and also some germline data like whole genome sequencing could stay with the patient for throughout their life, right? If you can manage the genomic data properly, a patient could be benefited, or an individual could be benefited, from these, throughout their life, So I think this is one of the uniqueness of genomic data is, it's beyond the condition you have at that point. It goes carries along the future of the life. So again, overall, I think we are expanding beyond clinical labs or EHRs. We are going for payers, pharmacies, and public health organizations, and also the ultimate goal is integrating genomics into real world patient care and treatment decisions. That is one of the key elements we are able to demonstrate. I think with that, I would like to pass on to Bob Dolan. He'll be speaking about next 2 use cases, which we kind of just started. Thank you all.
Yeah, thanks. Thanks. I think before I start talking about these 2 new use cases, there's just a couple of comments. First off, really great to see the folks on the participant list, a lot of friends and colleagues, some of which it's been years since we've even caught up. And I remember, we've been talking about where are we going to go with genomics? How are we going to achieve these objectives? How are we going to integrate genomics into cancer care? And I know that a lot of the folks on this call have been instrumental in getting the world, the United States to where we are today. And what we're trying to do here is leverage the CodeX accelerator and those groups that are interested in advancing FHIR-based strategies to achieve a high degree of genomic EHR integration as one of the strategies for improving cancer care. And in some cases, we're not necessarily the brilliant upstream researchers that make all the discoveries, but we feel like we're the actuation arm, the integration arm that brings that data ultimately to the point of care. So anyhow, thanks a lot, everyone, for being here. I'm not going to be blabbing for that long, so we're going to have plenty of time to hear from the audience. And in fact, looking at the audience, there's some folks here like Arthur Herman, who was instrumental in standing up GenomeX. And when I'm done, I'm going to invite some of the participants that also would like to chime in. Now, my last remark before I get started is to just digress a little bit about the CodeX development process so that I can explain where these 2 new use cases, the molecular tumor board use case and the pathogen genomics, are in the big trajectory, and why this is such a perfect time for you all to be learning about these use cases. So the CodeX use case development methodology begins with what they call the ideation phase. And that's when a group of like-minded people come together and say, we have an idea. We believe that we can put our heads together, put our resources together, and make that idea come to fruition in half the time, or in a quarter of the time, or in 2/3 of the time. And that's what a FHIR accelerator is, right? It's all of us getting together, identifying priority use cases, and getting that stuff done more quickly. So the ideation phase is where we kind of describe at a high level what it is that we want our committee to do. And that then sets the bar where we have now created these 2 new committees within CodeX, the molecular tumor board use case and the pathogen genomics use case. From ideation, we then go into discovery. And the discovery phase lasts whatever, I'm just going to say roughly 6 months. And it's during discovery phase that the committee kind of gels on what are the things that we're actually going to do? What are we as a committee going to roll up our sleeves and actually do? And in these 2 use cases, and I imagine a lot of other use cases, we're coupling our discovery phase to our outreach phase, trying to make sure that as these new committees have formed, in addition to trying to gel on where do we want to go, we're also in parallel bringing in new members. And so I'm going to present to you some ideas about where these 2 use cases might go. But what I'll tell you is over the next 6 months, depending on who decides to join these use cases, it's that community that's going to really shape where we go. And I see some of the participants on this list, and I know some of the things that we've debated. And all I can say is we have an opportunity here through CodeX, through these 2 use cases, to really dig in and explore some of these opportunities. The molecular tumor board use case kind of came about because we initially had a use case called operations, and that was an attempt to expose the patient's entire genome for advanced clinical decision support. And one of the pilot projects under operations was molecular tumor board. And so over the course of the past 2 years, we've been doing quite a bit of experimentation with molecular tumor boards. And then we began to realize, molecular tumor board is kind of like, what is it, outgrowing operations, and really is crying out for a use case of its own, because what we're seeing is that there's a tremendous growth across the globe of people standing up molecular tumor boards, taking the classic tumor board and beginning to infuse molecular data to aid in decision making. And what this slide here shows is just some of the publicly available screenshots that we've been able to grab. But it's, it's like almost every week you're seeing a new article in the literature from some other country is describing their national trend in molecular tumor board, or some university is describing where they're going. So again, we're super early in our discovery phase. We're super early in our outreach phase. So everything I tell you about where we're going to go with molecular tumor board and where we're going to go with pathogenomics, take it with a big grain of salt. We're going to work that out over the next 6 months. What we're thinking that we want to do with molecular tumor board is gather together an ecosystem of people that are involved in the molecular tumor board, whether that's the lab that's creating the data, whether that's the vendor that's creating the molecular tumor board application that's visualized by clinicians, getting these people together and trying to tease out what are the common challenges that we all have. We all have challenges with dynamically annotating variants with therapeutic implications. We all have challenges with oncogenicity prediction. We all have challenges with the semantic representation of fusions, right? And so what are the challenges? And in a way, it's a little bit of a weird use case. I don't know, Susan, that we've ever kind of talked about this, but it's a little bit of a weird use case for CodeX in that it's almost at cross direction to many of our other use cases, and that we already have a clinical trial matching use case in CodeX, right? We already have a data exchange use case. And so the molecular tumor board is going to be identifying now from almost, I guess, from a product deliverable perspective, what are the key things that we need? And no doubt, the molecular tumor board use case is going to be overlapping and integrating with many of the other use cases in CodeX. But to me, that's really a great thing, because wherever we decide we need to go with the molecular tumor board use case, we're going to find subject matter experts within CodeX to kind of help us. So if you could go to the next slide, please. So again, where we are in the process of molecular tumor board and pathogen genomics is we've developed our ideation page, and we're now in discovery page. This slide here is a synopsis of our ideation page. Some of the problems that we want to solve with molecular tumor board is, there might be some, what's the classic teaching is that evidence takes 7 years to get into clinical practice. And, obviously that's, in some cases an overstatement, but it's also ridiculous, right? Why isn't evidence immediately available through clinical decision support that's presented directly to the clinician? So you have these great therapies out there, and yet patients are potentially dying or having their lives shortened only because the doctor didn't know about that therapy. And molecular tumor board represents an opportunity where we can deploy decision support, we can deploy recommendations directly to the frontline clinician to help guide their decision making. And so one of the goals of all of the molecular tumor board applications that we see out there and that we recognize in this use case is that a molecular tumor board application can surface actionable variants and corresponding treatments and help guide the clinician to providing the optimized therapy for this particular patient. We also know that at least the literature reports how much time is required to prep for molecular tumor board, the curation time. And so we can explore developing automated annotation pipelines. We've done some work already in collaboration with GA4GH using their knowledge representation standards. So we know that we can decrease manual curation time. We know that we can prompt clinicians with available therapies. We can automatically deliver therapy recommendations from professional guidelines. We can automatically alert clinicians of available therapies based on public or commercial knowledge bases. Theoretically, we can help prioritize amongst available therapies and predict oncogenicity of novel variants. In fact, the ClinVar now supports submission of not only germline variants, but also somatic variants. And we can compute the oncogenicity prediction required so that molecular tumor boards can even submit novel variants directly to public knowledge bases, simplifying the whole learning health model. We can perform clinical trial matching, and a lot of the early members in the molecular tumor board use case are really pushing us to explore the learning health model, where the molecular tumor board not only is guiding the clinician, but is also capturing the actions of the clinician and standardizing those actions and putting them into a repository that can be aggregated with data coming in from other molecular tumor boards, where we can then glean insights, push those insights into a knowledge base, where that knowledge base is then tied to automatically informing the clinician about recommendations, thereby just basically blasting out of the water this time delay that's typically described for translational research getting into clinical practice. So again, we're early on. So what I'm going to tell you now is where I think the molecular tumor board use case might go, but I really hope, because I see that there is a boatload of cancer experts in the participant list. I hope at least one or two of you decide to come on board, help guide us, share your wisdom with us. But I'll give you an idea now of where I think we might go with the molecular tumor board. And at a high level, what we're going to do is engage a community of people that have some involvement in molecular tumor board, whether that's the lab producing the data, whether that's the vendor creating the molecular tumor board, and tease out what are the FHIR-based solutions that if we advance will raise the bar for all of the people who are somehow implementing molecular tumor board. The big items that we've discussed so far is data interoperability and standardization. There's a lot of, and in fact, as Srikar mentioned, biomarkers, for instance, there's inevitably going to be a lot of cross-fertilization between the work that we do and the GenomeX data exchange use case, because a lot of the data types that are important for cancer care haven't yet been standardized through the FHIR Genomics implementation guide. But there's also some things that we may do to, for instance, collaborate with GA4GH. We've seen some interesting work in knowledge representation that we believe that we can leverage to enhance the ability to do a variant annotation. And we think that if we solve some of these problems, it's going to benefit anyone who's developing a molecular tumor board. So again, what I'm thinking that we're going to be doing in the molecular tumor board use case is over the next couple of months, in parallel with doing outreach, is enumerating here's all of the different things, the pain points that we all share, and then we'll prioritize those. And then in 6 months from now, as we get into the planning and execution phase, we will have prioritized here are the things, the common pain points, and these are for instance, the two that we're going to focus on first, and then we're going to address these, kind of next. So that's kind of sort of where we'll go with molecular tumor board. And if we could go to the next slide, the pathogen genomics. This is, in my career of doing informatics, this one has arisen in a way completely like I've never seen before. Like we're constantly being inundated by pathogen genomics experts that want to solve problems. And the way that this kind of came to be was some of the pathogen genomics experts from Simon Fraser University in Canada came to HL7 and came to the clinical genomic group and said, look, pathogen genomics is really evolving into not only a public health challenge, but also an individual patient care challenge. And we're beginning to understand how we can use genomics to calculate antimicrobial resistance. In some cases, in theory at least, we can do it even more rapidly than the lab can do it using the standard methods. And if we can achieve the ability to communicate antimicrobial resistance in FHIR format, or if we can achieve the ability to literally send viral sequences or antimicrobial genes in FHIR, not only are we enabling patient care scenarios, but we're enabling public health scenarios. And so this group came to HL7, and we were kind of figuring out, well, what's the best home for this group? CodeX was like, hey, welcome, please come here. We'd love to help see if we can accelerate this use case. Where I think we're going to go with pathogen genomics, and again, this group is also just in ideation phase. We're just entering into discovery phase. So take everything I'm about to say with a grain of salt. But where I think we're going to go with pathogen genomics is to develop a new HL7 validated implementation guide that describes how to communicate pathogen genomics data from the lab to the electronic health record. It doesn't look like our initial use case will be public health focused, but we've been working with the public health community. We met with the Helios Accelerator, we met with the public health chairs from HL7, and they'll oversee our work with the notion that if we can formalize and standardize the data that's flowing from the lab to the EHR, it should be a straightforward step to then roll it up into aggregate to enable public health reporting. So we thought that we would focus on the patient care use cases first. Also, within HL7, there are some new FHIR resources being developed. In particular, there's the molecular definition resource being developed targeted for FHIR R6. And this is a resource that's naturally lends itself to formalizing the structure of a virus or the sequence of a virus. It's designed to communicate sequence data. So where I anticipate that we're going with pathogen genomics, but again, take it with a grain of salt, is we're going to build an implementation guide that defines how do you communicate pathogen genomics data from the lab to the EHR. And I would say if there were no such thing as a FHIR accelerator, a project like that would probably take 3 years, and you potentially groan and like, oh my gosh, 3 years, and maybe 3 years is right, because it's going to take a while for the country or the world to begin developing the technology to report that. But I think it's reasonable that with a FHIR accelerator, we can cut that down to 2 years. I don't think we want to cut it down to 1 year because there's too many people that have to adopt and have to implement. But I think that we can dramatically decrease the amount of time required to produce something like a new implementation guide and ballot it and publish it through HL7. We did find out that pathogen genomics is a nascent domain, not only in FHIR, but in HL7 through our discussions with public health standards. So I think that this may be the direction that the group wants to go. But again, through outreach, we may bring in some people that feel strongly and sway the group into going in a different direction. And so I would really invite those of you that have an interest in molecular tumor board to poke your head into the use case and just see what we're doing and offer us some guidance. And likewise, poke your head into the pathogen genomics use case, just see what we're doing, give us some of your expert guidance, or tell us some of your burning use cases to kind of help shape where we ultimately land on when we decide it's time for us to enter into the planning and execution phase. So with that, maybe just one more thing I can say, which is this whole transition in genomics under CodeX was thanks to the PMO, the Project Management Office. And for those of you on this call that don't know, we have an excellent PMO, and I just want to thank them for helping to really guide us in reshaping the GenomeX domain.
Oh, thanks, Bob.
That's it.
I have to agree with you there, Bob. And for Bob and Shrikar, your dedication and your passion and what you have done that I'm learning about right now is fabulous, right? And what we can do moving forward, I think the sky's the limit, and we have some great people. And so I guess with that, our ask from you all on the call is very simple and strategic, of course, right? We invite you to get directly involved in the genomics use cases, whether you bring clinical, technical, policy, payer, or even understand that you being whomever you are, you're also the patient, and you have patient expertise and experience. And so that insight alone can influence the direction and success of our work. So there are clear opportunities to align with your organization's priorities, from patient care to data infrastructure to reimbursement strategies. We have this, like I said, the sky's the limit. So as we move from strategy to pilot to scale to we're actively looking for collaborators to help shape practical standards-based solutions that solve real-world problems across this ecosystem. And probably most importantly, GenomeX doesn't exist in a vacuum. So your engagement can help bridge GenomeX with the other CodeX use case domains, create a broader value across cancer, cardiovascular health, and beyond. So we look forward to moving this forward together with all of you. And we open up the last few minutes. Shrikar and Bob said a lot, and there's a lot out there for us to talk about. So if there are any questions, we kind of open it up to you guys now.
Well, thanks, team. I would invite folks to post questions into the chat or to raise your hand, and I'll call on you. There's enough folks here that we don't just want to have folks coming off mic without an invitation. While we wait for questions to arrive, just want to address some questions that were in the chat about the deck and the recording. Both of these, as in other community of practice events, will be shared via our Confluence site. The link is in the chat. And so yeah, later today, I expect we'll publish the deck and the recording and some notes. So you are all welcome to share these with your colleagues and friends and organizations as you see fit. Because everything, as much as we can anyways, everything that we do here at CodeX is meant to be public and is meant to be for the public good. All right, Rebecca extends her regards. Any other questions from our audience? Or Bob, would you like to ask a question of any of our audience, or would you like to invite anybody to speak?
Well, I want to put Arthur on the spot.
Yeah, you threatened to do that earlier.
What's that? Yeah.
You threatened Arthur earlier, so perhaps he'll take you up on it.
Arthur, are you there? I don't know. He's on mute. He may have had to step away. So if not,
I am here.
oh, hey, I just wondered if you wanted to say a few words.
No.
Okay. All right.
I think you guys, I'm being facetious, but I think you did a very good job of presenting everything. I guess I will ask a question, Bob, because you put forward for pathogen genomics a particular use case. At the same time, you're saying that's not defined. And the use case you said was building a full implementation guide. And I guess I'm wondering why you decided to put that one forward when there actually are 3 or 4 that are strongly being debated, at least from my understanding, unless some things happened since I last talked to you.
Oh, it's just my philosophy in standards development is always have a straw position. It's easier to go into a room with a straw position and change it than to go in and saying, we have all these options, what do we do? That's all.
Okay. Got it. Yeah, I would just, for the audience, I would just say, really where we are is probably, I'm going to be grand and say 5. We've got 5 different ideas that resonate with lots of people, and the next 6 months will be finding out who wants to be involved, what their priorities are, and eventually then defining which of those becomes the initial priority. That's it.
Thanks, Arthur. Yeah, we are, we are very proud of and invest heavily in our use case methodology. And so use cases go through these particular phases and through these gates. And over the past 5 years of CodeX life and experience, we have found that these have been refined, but that these work really quite well. And so we do like to continually iterate and improve. And for instance, as Kelly pointed out earlier, our new GenomeX leadership team is an iteration on previous governance models. It's meant to be a careful and slow improvement. And all of our records are shared publicly. Some links are in the chat now for some of the records of this team and its work. So if you have any questions or comments about CodeX processes and operation stuff, that's where the PMO, my colleagues and I are here to answer your questions, and our contact is there on screen. I think I'll pass it back to Kelly to close us out. If there are any other questions, you have just a moment left. Please put them in the chat. Over to you, Kelly.
Yeah, with that, if you have questions and we run out of time, feel free to reach out to any of us. Our emails are listed here. We welcome anybody to even stop in and hear and see what we're doing. We can arrange that to get you more involved. So really, we're willing and open to get you involved any way that we can, and we definitely appreciate having you on the call with us today. So thanks very much. And again, thanks to Shrikar and Bob for all the amazing work you have done and all the work ahead of us that we're going to figure out how to get to. So appreciate you. And James, if you're still on, everything that you did, it's amazing, and you have a great following and a lot of great comments that I've been hearing as I'm learning the ropes. So thank you for all you did to help me and all you did to help the group.
Of course. Thanks, Kelly.
Thank you all. Bye.
Very good. I will now stop the recording and wish you all an excellent day and a great weekend.
Thanks, everybody.